Hello everyone. So I'm going to go through the Unit 12 test, and in this I'd like you to think about whether you have got the knowledge, whether it's exam technique that you've fallen foul of if you haven't got the marks, or whether it's understanding. For exam technique, obviously that's about experience, so do clock it. For knowledge, often it's about working harder, have you got the detail in? And for understanding, it's really important if you haven't understood something that you take responsibility for that and you make contact with me so I can help you. Okay. So for the first question, DNA is genetic material of human cells. Describe where DNA is found in a human cell. Well, the first thing to say about this is look at this. It's 2 marks. So a lot of people put nucleus. That's not going to cut it, guys. You're not going to get 2 marks for nucleus. So nucleus is 1, but it's on chromosomes. Okay, in the nucleus. All right, and so exam technique wise, you should be thinking if that's 2 marks, I'm looking for something else other than nucleus. And then chromosomes is knowledge. You've got the this here is a sequence of DNA which makes a chromosome. These are the base pairs A, T, G, C. Okay, and this double strand of DNA is going to be on a chromosome. Okay. So, when a cell divides by mitosis, the new cells are genetically identical. What causes the cells to be genetically identical? Okay, so the DNA in mitosis is cloned, or a clone is formed. And so, if we look at the mark scheme here, you can see that the answer is the DNA chromosome genes are replicated, copied, multiplied, doubled, duplicated. Allow DNA is cloned. Okay, ignore same DNA chromosomes genes unqualified. So we're looking at the fact that there's 2 genetically identical copies in mitosis, which would lead to the DNA being identical. Okay. So when a cell divides by mitosis, that's the next question. Many genes have different forms called allele. A person has polydactyly, extra fingers or toes. Polydactyly is caused by a dominant allele. What is the smallest number of copies of the dominant allele for polydactyly that could be found in a body cell of a person? Okay, so this is about knowing about dominant and recessive. So you get 1 chromosome from your mother, okay, and you get 1 chromosome from your father, and they both donate an allele. So if you have a gene here that is, say, for eye colour, and it is dominant, like brown, you only need to have 1 brown there for the phenotype, what you see expressed to be brown. So if you had another, it would be a bonus. Okay, but if it was a small b there, the brown would still come out. And so you only need 1 dominant allele for that dominant allele to be expressed. In 1.4 here, we've got how many copies of the recessive cystic fibrosis allele we need for this person to have cystic fibrosis. Well, because cystic fibrosis is a recessive allele, you would need both of the alleles to be recessive. So the answer here is 1, and the answer here is 2. Okay. A burglar broke into a house. Everybody got this right. That's B. Well done. The colour of the mouse's fur is determined by a single gene. Different version of the gene known as alleles. Explain how a gene codes for a characteristic. This is actually tricky. And you've got your section of DNA here. And you've got the bases, the ATGCs. And basically what happens is that that DNA codes for amino acids at the ribosomes, which then go to proteins, and that cross-references with digestion where proteins are broken down into amino acids form proteins. That's a tricky question, and that fully is understandable why you wouldn't know that. Okay. Describe the difference between dominant and recessive alleles. Now this is knowledge, guys, and I went over this again and again about the words we've got. So dominant is either or, and recessive is only. And how does this fit in? So a dominant allele is expressed in either heterozygous or homozygous condition, and recessive is only expressed in the homozygous, okay, or when there's 2 alleles there. It's a bit similar to the earlier question, actually. Okay, so really important that you know those definitions. Describe the difference between genotype and phenotype of the mouse. Well, once again, this is knowledge of learning those definitions that we created on the PowerPoint. So the genotype is the 2 alleles that are found at a gene. So if this was eye colour, for example, and you had big B and small b, the genotype would be big B, small b. The genotype is the alleles found at a gene, and the phenotype is the characteristics expressed that you can see. So in this situation, the phenotype of this genotype would be big B, small b. Brown is dominant, so that would be brown eyes. That would be the phenotype. Okay, so those 4 marks were for learning definitions. Okay, and that's knowledge and hard work. 2.4, the allele for black fur, B, is dominant allele for brown fur. 2 heterozygous mice, so you need to know that heterozygous means 2 different alleles. So we would have big B, small b, and big B, small b. And most people got this right, guys. If you multiply this out, you get big B, big B, big B, small b, big B, small b, and small b, small b. Now, you get a mark there for, let me just show you how these marks. So you get a mark here for getting the correct genotypes, big B, small b, both times. You get a mark for separating them out for the cross. So the 2 options that big B, small b could donate are a big B or a small b. So you've already done 2 marks. And then if you get the cross right with the big B's, if you get these right, you get another mark. So you're on to 3 just by doing the cross. Now, lots of people, though, didn't read the question and didn't follow my advice about what you need to do in these crosses, which is to give all the information as well. So the question says, perform a genetic cross to show the expected phenotypic ratio. So many people didn't show the ratio. Okay, and the way you do that is by stating the phenotype. So the phenotype here of big B would be black. The phenotype here of big B, small b would be black, because black is dominant. Big B, small b, black. And small b, brown. So you get a mark for stating the phenotypes. And the last mark is for then giving the ratio. And the ratio is 3 to 1. And so specifically for brown, it's a 25% chance. Okay, so lots of people got 3 there, but didn't mention the phenotype and didn't mention the ratio, and that was in the question, so that was quite careless. Okay, moving on. Over a series of litters, the mice had 18 offspring, 13 had black fur, and 5 had brown fur. Explain why these results are different, the expected calculate. So if you're talking about a ratio of 3 to 1, ideologically, 13 to 5, you can see is not quite 3 to 1. If it was 3 to 1, it would be 15 to 5. And so what this is asking is, why is this number different from 3 to 1? Okay, and this is a really good one to use the mark scheme for, because this could be a question that comes up and there's a set answer for it. Okay, let me show you what it is. So it's here. The calculated 2.5. The calculated ratio is a statistical prediction. The actual results are dependent on which sperm fertilizes the egg, or the sample size was too small to approach the expected ratio. Okay, so if we go back here and just think about explaining this. Okay, so if you've got a sperm and an egg, okay, these sperm and egg are going to have different alleles in them, okay, and it's completely random. So you wouldn't expect all the time to get the exact ratio, and you can describe this by, in the lesson I was talking about throwing a head and a tail. If you throw a coin 100 times, you're not going to get exactly 50-50, although statistically you should. In real life, the random nature of it means that you're likely to get slightly off from that. So it might be 45, 55. And the second part of that answer is that if you did it 1,000,000 times, though, you would expect it to be very close statistically. So 500,050 and 499,950. Okay? So, tricky question there, and that's just a good one to learn the answer to. So Mr. and Smith both have a history of cystic fibrosis in their families. Neither of them has cystic fibrosis. Mr. and Smith are concerned that they may have a child with cystic fibrosis. So this is draw a genetic diagram to show how they could have one. So neither have got it. So they cannot be, so they're looking for A. So they cannot be AA. Because if they were AA, they'd have it. Okay? So they cannot be AA. So we can do away that. Now, if they could have a child, they both must be able to donate a small a. So their genotype must be AA and AA. Let me explain that to you. If it was, let's say it wasn't AA and it was big A, okay? When you do a cross of A, you get that. Now, they're asking to show that they could have a child. Well, you can look at this here. Every single one of those phenotypes that has been created has got a big A in. So there is no cystic fibrosis there. And the question is asking, how could you show that one could? So if we cross AA times AA, they can both donate the small a to create a double recessive small a and therefore cystic fibrosis. But they want for 3 marks us to show this. So we use a Punnett square to show A, big A, big A, big A, small a, big A, small a, small a, small a. There is the double recessive, which would have cystic fibrosis, and then you need to label that as the cystic fibrosis child. Okay, and that gives you the 3 marks. Some people just did the Punnett square and didn't label the child. Okay, so you need to have gone on to do that. So 3.2. 5 eggs will be removed from Mrs. Smith's ovary while she's under anaesthetic for embryo screening. The eggs will be fertilised in a dish using Mrs. Smith's sperm. The embryos will be grown in the dish until each embryo has had 30 cells. 1 cell will be removed from each embryo and tested for cystic fibrosis. A suitable embryo will be placed in Mrs. Smith's uterus and she may become pregnant, and only unsuitable embryos will be destroyed. Suggest why it's helpful to take 5 eggs from the ovary, not just 1. Well, that's quite straightforward. That increases the chance of having a child that doesn't have cystic fibrosis. Now, evaluate the use of the embryo screening in this case. Remember to give a conclusion to your evaluation. Some people didn't read the question properly and didn't give a conclusion. So what does evaluate mean? Well, evaluate means give the pros and the cons and then come up with your own conclusion. It doesn't have to be a correct conclusion. Even if, as long as you have said, this is what's good, this is what's bad, and this is my conclusion, you're going to get a mark for the conclusion. So if we look at the mark scheme here, you can see the kind of content that you could have put in, okay? So advantage, and once again, this is really good to learn because this is the exam board's answer for this question on the advantages and disadvantages of embryo screening. So advantages are great in a certain chance of having an embryo with cystic fibrosis. Okay, so not having a child with cystic fibrosis. Okay, it's if you stop a child with cystic fibrosis coming into the world, it will be very expensive to bring up, and that's a problem. So if you don't have that child, then that's an advantage, and it would also stop the allele for cystic fibrosis not being passed on to future generations because that child would have a double recessive AA, small a, and that means if that person had any children, they would definitely donate the recessive A allele. The disadvantages you could have had is reasons of dangers due to an operation, ethical or religious issues, high cost, possible damage to embryo. And then a conclusion is just saying, I think they should have it because, or I think they shouldn't because, okay? And that's 4 marks for that, okay? And the last question, guys, is this very, very tricky question. which is thrown into, this is like a grade 9 question. And this basically is about osmosis. If chloride ions move out, okay, then water will move by osmosis from an area of high water concentration to low water concentration, okay, and through a semi-permeable membrane by osmosis, and that will make the mucus thicker, okay. So that, once again, is a very hard question, and one which I wouldn't necessarily expect you to get, and it wasn't even in this topic, so it was a real left field question which the exam would use to differentiate students who are really thinking outside the box. Okay, so guys, please look at your answers. Was it knowledge? Was it exam technique? Was it understanding? Let me know if I can help at all.